De-extinction is a new and probably the most fascinating scientific project at the moment, not only in the field of conservation biology and genetic engineering, but from the point of view of science as a whole. The aim of de-extinction is to bring back extinct animals and reintegrate them into their former ecosystems. One of the species soon to be resurrected is the legendary marsupial, the Thylacine, or Tasmanian tiger. I have described the process of its de-extinction in one of the videos on this channel, which you can watch by clicking on the thumbnail. As expected, this topic proved to be very popular with the audience. But this time I would like to remind people that watching a few minutes of video is simply not enough to get a real and critical view of this matter. First of all, genetic engineering is one of the most difficult projects ever undertaken by science or mankind, comparable in fact to the conquest of space. It requires not only cutting-edge microbiological knowledge and an understanding of how genetics works, but also a range of extremely expensive, perfectly functioning, most advanced technological equipment. Transferring the bare theory of genetic sequencing and reproduction to modern instruments is not to be sneezed at, which is why the very greatest minds of our time are working in the field of genetic engineering and in particular, de-extinction. The methods they use are almost impossible for most people to understand. Fortunately, however, there are now enough explanations and videos on the internet to make these remarkable historical milestones in science comprehensible to everyone. But to provide a simple and straightforward explanation, it is necessary to leave out some details and intricate particulars which are otherwise absolutely crucial to the process. Because ordinary people never find out about these small important things, they imagine processes like de-extinction in a rather simplistic and facile way. Often they even doubt it. For example, my video has also been met with the reaction that this comeback is a waste of money, as well as the accusation that the creature created will not be a Tasmanian tiger at all, and of course the ever-present question of why it will take so long for a Tasmanian tiger to be brought back. So I decided to make a video specifically about the reasons for the slow, or rather cautious, pace of this project. I will try to explain the important details of the resurrection biology process as clearly as possible, using the Tasmanian tiger as an example, so that no one will be any longer confused. Yes, we are going to get even deeper than in my previous video. But before I start with the first reason, I would like to ask you to subscribe to my channel. It really takes a lot of time and even more effort to make content like this, because a quality explainer video requires a lot of research, gathering credible data and putting it together into a comprehensive presentation. If you would like to help me in any way, or thank me for the video, I would love it if you would subscribe. It's free and so quick, it couldn't be easier. So once again, please go ahead and do it. Now, let's start with the reasons. Biologists and biotechnologists are still struggling with some of the problems at the reproductive level of the Thylacine de-extinction. It is clear that a Tasmanian tiger embryo can only develop successfully with the help and cooperation of a surrogate mother. Scientists have already decided that this will be a female fat-tailed dunnart, as this tiny marsupial is Thylacine's closest living relative. Over 95% of their genomes are identical. Creating a so-called marsupial wolf from a marsupial mouse that is more than 100 times smaller may seem bizarre, but in fact the size difference is the least of the problems. All marsupials are extremely small at birth, about half the size of a pinky nail. The situation is complicated because it is difficult to find a way to prepare a surrogate female to survive and carry the pregnancy to term. There is even a small chance that her body will reject the embryo and not feed it, considering it unnatural. Looking back in history, the first attempt to revive an extinct animal was in 2003, when scientists implanted a goat egg with a nucleus from the extinct Pyrenean ibex into a surrogate goat mother. In this case, there was no problem with adoption, but the nucleus was taken directly from the tissue of the last living Pyrenean ibex, so it was purely natural in origin. But in the case of the Tasmanian tiger, it is an artificial nucleus created in the laboratory, so placing the embryo in the uterus of a fat-tailed dunnart could be questionable. We cannot replace the mother with some kind of stimulating device to help the embryo grow, because building such a tool would cost millions of extra dollars and prolong the wait even further. But assuming that the mother does manage to keep the embryo, that it does settle comfortably in her system, and that it is then born, what then? Clearly, the surrogacy will have to end as soon as possible, as the female will not be able to care for the pup for long. It will outgrow her in a matter of weeks. Then there is the question of how the newborn will fare and whether it will survive weaning at all. To recall the story of the Pyrenean ibex, a female kid born to a goat in July 2003 died 7 minutes after birth due to a lung defect. In other cloning attempts, the animals were also very fragile and did not survive long. Scientists will have to plan very carefully how to provide the best possible care for Thylacine Joey after birth, 24 hours a day, to give it a better chance of survival. After all, the Tasmanian tiger comeback attempt is undoubtedly the most expensive animal genetic engineering project to date, and it is backed by many famous investors. Some sources say the final cost could easily reach $50 million. It would be a major defeat and a huge financial loss if the project ultimately proved to be a failure. The second problem is, yes, we have a genome, but not all genomes are the same. In fact, the team of scientists I talked about in the video above were able to construct or assemble a high-quality reference genome after extracting DNA from 100-plus-year-old pouched young specimens stored in ethanol. Explaining and understanding what a reference genome is far from straightforward, but to simplify it as much as possible, it is a representative example of the set of genes in an idealized individual organism of a species. If this sounds confusing, don't worry. The reference genome is useful to biologists for comparative research, but it cannot directly contribute to the return of the tiger, because it is enough for us to create only one single specimen. That would be a drop in the ocean compared to the goal of establishing a stable population of Tasmanian tigers. What we need for a stable population is a large pool of diverse genetic material so that the animals created can reproduce. That is why experts at the University of Melbourne have said that the most important step towards de-extinction is to create a functional genome. Using the current reference genome, the first urgent step is to create more new so-called embryonic stem cells. And what are these? They are the cells from which we can then make reproductive animal cells, also known as gametes. This means that by making and storing stem cells, we can essentially provide a wide variety of sperm and new eggs to create the genetically diverse Tasmanian tiger embryos. So when we combine the reference genome with the stem cells, we get the functional genome that we need if the Thylacine de-extinction project is to be prospective and successful in the long term. The final and probably most difficult problem to solve is the planning and organization of what we will do with the genetically engineered offspring. In practical terms, how we will manage their growth, development and success. The scientists keep saying that their rescue mission goes far beyond the birth of the first Thylacine Joey. The ultimate goal, of course, is to begin reintroducing this lost keystone species, with the help of conservation organizations, to specific areas of Tasmania, and probably parts of mainland Australia, so that it can regain its vital role in the ecosystem. But there is still a long way to go. The first generation of Tasmanian tigers to be introduced will certainly not be ready for release into the wild. They will need to grow up in a controlled environment where scientists can continuously monitor their vital functions. As I mentioned earlier, there is a high chance that they will be quite fragile due to any deficiencies or developmental defects in certain organ systems, particularly the respiratory and circulatory system. But even if this shortens their lifespan, it will not be a total defeat for scientists, but an opportunity to see where the mistakes are and to correct them, possibly in the very next generation. It will probably take several generations before we get 100% healthy specimens suitable for reintroduction. But even that will not be the final victory. Even when, or if, the first Tasmanian tigers are released into the wild, they will need to be closely protected and monitored, at least in the early stages. Their uniqueness, origin and the investment that has gone into their creation will undoubtedly make them the most valuable creatures of the moment. They will need to be strongly protected from negative external influences, as well as possible threats in the form of poachers, until a sufficiently large and stable population can be established. An effective framework for achieving this goal requires hundreds of comprehensive plans, from establishing suitable breeding sites, to ensuring security conditions, setting up technology to help us monitor specimens, working closely with local governments, communities, numerous wildlife and ecosystem conservation organizations, and so on. As this involves a lot of complicated bureaucracy, obtaining various permits and, above all, the need for even more investment, things are moving rather slowly. Ordinary people often grumble about this, assuming that the process is being unnecessarily delayed, that too much is being hidden from us, and that we are basically being made fools of. In fact, we should be more tolerant and realize that something like the de-extinction of the Thylacine has never been done before by scientists with any species, so slower and more cautious steps should be quite understandable to us. To conclude this video, I would like to talk to you about what we can learn from the process of the Tasmanian tiger's de-extinction and how we can benefit from the lessons we have learned. Surely the most important lesson is that genetic engineering cannot be our regular tool to solve the problem of continuing species extinction. We cannot rely on the fact that every animal that we wipe out of the world through selfish and reckless behavior can be quickly created in the laboratory. As we have seen in the case of the Tasmanian tiger, this is a lengthy and extremely demanding process, requiring enormous financial resources in addition to the scientific work, so we will not be able to afford it very often. We should therefore remember that protecting nature and wildlife from pollution, habitat loss, hunting and global warming is by far the most effective, economically viable and best way to combat mass extinction. So while we await the return of the super predator to the old-growth forests of the Tasmanian wilderness, let us first and foremost ensure that in the meantime we do not put any more stress on the environment and cause another of its remarkable endemic animals to disappear and be doomed whether or not we decide to bring it back to life.